Hey guys, welcome back to the screencast. This is for AP Biology. We are specifically in a unit on metabolism and enzyme activity. And so this particular screencast is going to focus on textbook section 6.4, enzymes speed up metabolic reactions by lowering energy barriers. So first of all, we want to know that enzymes are referred to as biological catalysts. And a biological catalyst is any kind of chemical agent. It's basically going to speed up a reaction, but it's not going to be used up, or it's not going to be consumed, which means it can be recycled and reused over and over and over again. So every time an enzyme works in a particular reaction, the enzyme is not going to be changed by the reaction itself. Now, an enzyme can change shape. We'll talk about enzyme denaturization a little bit toward the end of this screencast. But the enzyme again can be used over and over and over within a reaction. An enzyme is a macromolecule specifically. It's typically a protein. So we know our macromolecules are carbs, lipids, proteins, nucleic acids, and enzymes are typically proteins. We've got an example here. One example of an enzyme is going to be sucrase. Remember, most enzymes end in ASE, and sucrase has a specific substrate that it works on called sucrose, and that ose is a sugar. So the action of this enzyme, it says it catalyzes, so it speeds up the hydrolysis. So hydrolysis, remember, is that breakdown reaction of sucrose. So just a quick review, metabolism involves catabolism and anabolism, so that's both the buildup and breakdown, and enzymes speed up those reactions. Okay, here's a picture of the example that was just given related to hydrolysis. So you'll see this is a breakdown reaction. Specifically, sucrose is being broken down, and this would happen naturally over time. It might occur, however, so incredibly slow that if this was needed to happen within an organism without this enzyme, it wouldn't happen at the speed that was biologically beneficial to the organism. So the enzyme itself is not what's responsible for the breakdown or the hydrolysis, but it is responsible for the speed of the reaction happening. So sucrose over here, this ose again, is a carbohydrate or a sugar. Specifically, it's a double sugar. You can see that right here because we've got two monosaccharides attached to one another. And all reactions are going to involve like the breaking and building of bonds. So the bond here between these monosaccharides is going to break. Bonds between hydrogen, oxygen, and water is going to break. And so through hydrolysis, the bonds that break over here are going to then, if you look on this side, are going to form new bonds over here with new parts, and you're going to end up with glucose and fructose, two monomers that were broken apart from this polymer, and water was split in order for hydrolysis to occur. So overall picture, sucrase speeds up this breakdown reaction. Okay, so let's talk about the activation energy. You're going to see this in graph form. You need to have an understanding of the concept of activation energy, or some people might call it E sub A, same thing, but have an understanding of the concept and again be able to recognize it on a graph. So every chemical reaction, like I just mentioned, it's going to involve both the breaking and building of bonds, or bond breaking and bond formation. And so activation energy is basically the energy required to get that reaction started, meaning it's the energy required to start a reaction by breaking the bonds in the reactants. So if we went back to the picture before, the energy needed to initially break the bonds between sucrose, or between these I'm going to try to say monomers that are bonded together, the amount of energy required to do that would be referred to as activation energy. And then activation energy sometimes is supplied by like the surrounding heat within an organism, but heat isn't always the best source of energy in order for those bonds to break, because too much heat can denature a protein and cause a shape change where that active site isn't shaped properly for the substrate to fit in it. So enzymes are a more efficient way for that reaction to be sped up by lowering the activation energy needed. So on this particular graph, you've got a few points that are going to be emphasized, but the one right here, I want you to notice that whatever amount of energy has, well, let me start over. This arrow right here, once you notice, it's not starting from the bottom of the graph, it's not starting from the origin, it's beginning from wherever this line starts. So like the amount of free energy within this reaction to begin with, the peak of this hill is what we call that E sub A, that activation energy or energy of activation. This is the amount of energy needed, again, to start the reaction that in this picture shows a molecule AB and molecule CD going through a reaction together to form AC and BD. In this particular reaction, I can tell that this is a breakdown reaction because there is a release of energy. And so this release of energy, I'm going to see it looking like a slope going down further than where it started right here. And so if we go back to the analogy we've talked about in class, if I were to take money out of my bank account is going to go down, but that also puts money in my hand that can be spent. So in this particular graph, you're going to see a change in free energy, or what we call delta G, that's going to be less than zero. So it's going to have a negative change in delta G, or a negative change in free energy, but that also means free energy is available to do work, and that happens as a result of breakdown reactions or catabolism. Okay, we talked about this just a moment ago. Heat, can denature proteins. Too much heat can denature proteins. And we talk about denaturization of proteins, like we're talking about a shape change that alters the active site. And if we alter that active site and the substrate cannot properly fit, then the enzyme is not going to be functional. So when an enzyme is denatured, it might be temporary or it might be permanent. If I were to give you an example of like a permanent denaturization, if I apply too much heat, or maybe just the right amount for my particular situation. If I am to cook an egg, I'm denaturing that protein and it will never go back to the shape that it originally was. However, some of us denature proteins within our hair and we straighten our hair, and it doesn't mean that it cannot go back to a curly shape that it was before heat was applied, for example, with a straightening iron. So again, heat can be problematic in terms of providing energy in order to get reactions started without denaturing proteins. So organisms rely specifically on enzymes or catalysis to speed up reactions. Okay, so we talked about enzymes catalyze or speed up reactions by lowering the amount of energy needed to start the reaction, and that they're not going to be consumed in the reaction. And I'm going to come back to the second bullet. I want to show you the picture that goes along with this. So take just a moment to read that to yourself, and then we'll come back to it. So here's what, let me scoot to this picture. This is the picture I really want you to pay attention to. It looks like the last one that was shown to you, except there's two lines. And the one that is in red, this is going to represent the reaction with the enzyme. And the one that's in black, we'll look at that later. The one that is in black is going to be the reaction without the enzyme. So you can see that there is less energy required, or less energy of activation required when an enzyme is present. So kata means down. So we are lowering a catalyst, an enzyme is lowering the amount of activation energy required to get the reaction started. Again, this is a breakdown reaction. You can see that because of this drop or change in free energy, which is negative. And you'll notice this note, it says change in G is unaffected by the enzyme, meaning we end in the same spot. We also begin in the same spot. The only thing the enzyme is altering is the amount of activation energy needed to start the reaction. So I'm going back to that bullet I told you that I'd skip and come back to. It says enzymes do not affect the change in free energy. It's not going to have a change in the outcome of the amount of free energy that's available or stored as a result of the reaction. They only speed up reactions that would eventually occur without them. So the enzymes are not responsible for the reaction taking place. They are responsible for the speed of the reaction taking place. Okay, substrate specificity. So we know enzymes have particular substrates that they act on, that enzyme has a specific active site, and the reason it has the specific active site or the shape it is because of that domain or that portion of the enzyme has a specific fold of amino acids based on whatever properties those amino acids in that area have. So the amino acids might be hydrophobic or hydrophilic. They might be positively charged or negatively charged, but the interactions between those R groups of the amino acids on that tertiary structure of the protein is going to result in a specific fold or a specific active site in this particular spot of the enzyme where a substrate can actually bind. So the substrate is whatever molecule the enzyme is interacting with that binds together. And then when those two things come together, we call it an enzyme substrate complex. We've already mentioned this. Most enzymes end in ACE. So any molecule word that you see that ends in ASE, you're dealing with an enzyme, and all enzymes, well, almost all enzymes are proteins. And then again, an example, sucrase catalyzes the hydrolysis of sucrose, which you've seen already. The active site is the region on the enzyme to which the substrate binds. The enzyme specifically results from the fit between the shape of the active site and the substrate. That's what we were mentioning earlier. Enzymes change shape. Okay, so when we say they change shape due to chemical interactions with the substrate, you'll see a picture in just a moment where when the substrate binds, it kind of looks like it squeezes or hugs it a little bit tighter. So there's interactions, some bond formation that's happening between those two molecules. So there is an alteration of the shape change. I think of this kind of like putting a glove on my hand before I do a lab, that it's not already specifically designed to fit my hand, but once I put it on, it kind of conforms to the shape of my hand and the size of my fingers. This induced fit of the enzyme to the substrate brings chemical groups of the active sites together. So again, in this picture, you're seeing a substrate bind with an enzyme in a specific region called its active site. The active site is very important. And then once that binding takes place, you'll see that induced fit where it almost looks like it hugs that enzyme, and there's some interactions going on between the two. Okay, moving on. I'm going slow enough that if you want to pause, you can read some of the captions. Okay, catalysis in the enzyme's active site. So substrates are held together sometimes by weak interactions, like hydrogen bonds or weak interactions. The enzyme lowers the amount of activation energy needed to convert the substrate to the product. It's not going to change what product is formed. It's not going to change the amount of free energy that's available or not available at the end of the reaction. Again, it only changes the amount of activation energy needed to start the reaction, and it, in fact, always lowers it. Okay, after releasing the products, the active site is available to bind with more substrate molecules. So that goes back to our original point that the enzyme is not consumed by the reaction itself. So once the substrate binds and the interactions take place, let's say it's a breakdown reaction, so the substrate breaks apart and it releases, then that enzyme again is available to do more work. So here's like a quick picture where you see this enzyme, and then you'll see substrate AB that docks or binds at the active site, and then you can see that these have been broken apart, they get released, and then that enzyme again is available to do work on another substrate if available. Okay, I'm going to show this picture kind of in a little sequence here, which is exactly what we've talked about, so you can pause as you need to. You see those substrates are converted to products, the products get released, and then the enzyme again is not used up in the reaction. The active site is now available for more substrates to bind if they're available. Okay, so the active site can lower the activation energy and speed up reactions through these things that we've talked about, where it kind of positions the substrate correctly, and it provides a favorable environment for that substrate to either break apart or those pieces to come together. Okay, the rate of the enzyme catalysis can usually be sped up by increasing the substrate concentration. That's a really big deal. So this is going to be some of the factors that alter the rate of the reaction. So if I increase if I increase the concentration of the substrate, if I add more substrates, then I might speed up the reaction by causing more products to be formed, assuming there's an enzyme there. And then this term enzyme saturation. So when all enzyme molecules, let's say we have a fixed amount of an enzyme in a reaction, so we've got a certain amount of enzyme that we're not changing. When all enzymes in a solution are bonded with the substrate, the enzyme is saturated. So all the enzymes are busy doing work, enzyme is saturated. So when enzyme saturation is reached, the reaction speed can only be increased by adding more enzyme. So I would only be able to further speed up the reaction by adding additional enzyme to my system. Okay, and then we're going to end right here where it says effects of local conditions on enzyme activity. So factors such as temperature, pH, salinity, those things can alter the activity of enzymes. That'll be our next topic to pick up on, hopefully in my next screencast. So I'm going to pause it here. I hope, again, this has been helpful. I am going to actually show one picture. I think that I would like to go back to on this slide that we already talked about. I want you to notice how similar it is to a previous slide that we've looked at the very beginning. When we first had this topic introduced on metabolic pathways, you've got this starting material that's being converted from A to B to C to D, and enzymes sometimes are involved in these reactions, involved in the conversion of these molecules. And so then if we pick up to where we've kind of gotten to today, and you go back and you see, instead of just starting molecule A to B to C to D, today you see sucrose being hydrolyzed into glucose and fructose, and this enzyme sucrase is involved in that reaction. So you're seeing that metabolic pathway take place, and you're seeing the enzyme that is speeding up that reaction in the process.